Hi, everyone. Welcome to the PyTorch webinar, where today we're going to be talking about using PyTorch and DINOv2 for multi-label plant species classification. My name is Susan Kaler, and I am a developer advocate at Intel. Our speaker today is Murilo Gustineli. He is a senior AI software solutions engineer at Intel and is currently pursuing a master's in computer science at Georgia Tech, where he focuses on machine learning. His work involves creating synthetic data sets, fine-tuning large language models, and training multimodal models using Intel Gaudi AI accelerators as part of the development enablement team. He is particularly interested in deep learning, information retrieval, and biodiversity research, aiming to improve species identification and support conservation efforts. As we go through the webinar today, please enter your questions in the chat and Murilo will answer them at the end of his demo. Over to you, Murilo.
Yeah, thank you very much, Susan. Thank you very much. So let's get started. First and foremost, I'd like to thank the PyTorch Foundation for inviting me today to be presenting on this, and also to thank Intel for allowing me to share some of my personal research that I've been doing for my master's. And this is going to be a very hands-on type of webinar. I don't have any slides. We're going to dive right into the code. So I believe we have shared the GitHub repo that we're going to be working from today, which is going to be from my GitHub repo. And the name is going to be PyTorch/PlantCLEF. And all the code that I'm going to be running today is from this repo. And yeah, so basically, first of all, let's introduce the challenge that we're going to be facing today. So the webinar today, we're going to be covering this research that I've been doing at Georgia Tech, and it's called PlantCLEF. So if we click here, we're going to go to the Kaggle page. So basically, what is PlantCLEF? PlantCLEF is a machine learning competition. It's a challenge to identify plant species in high-resolution images of vegetation plots, basically trying to identify plant species in images. And this is a competition that happens for the live clay. I know that it's spelled as clef, but it's pronounced as clay because I believe it's French. So it's part of the Live Clay Lab, and the Live Clay Lab is part of the Clay Conference. And as you can see here, this is the official page on Kaggle of the competition. The PlantCLEF competition is also part of the, not only of the Live Clay lab, but also part of the CVPR conference. Well, part of the FGVC workshop within CVPR. So this is kind of the challenge. We're trying to identify multiple species of plants in images, and there's a lot of, it's really challenging because there's a big domain shift between the training data and the test data. So talking a little bit about that here, you guys can see that they're showcasing the main difference between the training data and the test data. As you can see, the training data is species or images of single plants. So there's only one species per image on the training data. On the test data, we have multiple plant species on this vegetation plot. So you may have one species up to 8, 9, 10 species. So I believe this one right here, I believe has 8 total different species. I'm not a botanist, so I cannot tell which species belongs to which class, but they actually did it here on the left. So we have all of those 1, 2, 3, 4, 5, 6, 7, 8 species here on the test data. And that's really the goal of the competition, is trying to identify all of the plant species that are on those vegetation plots here. And then they talk more about the evaluation metrics that the competition are evaluating their models, basically. So when you submit a prediction, they run through those evaluation metrics. And they also have a bunch of just information about how to submit your your submissions if you'd like to participate. I'll be talking a little bit about that later at the end of the webinar. But that's really what PlantCLEF is about, is this multi-species plants identification in images. And it's a very challenging very challenging problem to solve. And I'll be talking a little bit more about that. But for the workshop today, for the webinar today, going back to the repo, I have a README file with the entire setup for the code that I'm going to be running today. So I'm not going to be running through the setup today for the webinar, just to save some time, as we have a lot of content to go through. And I believe the webinar is going to be taking, at least the code portion that I'm going to be walking through, is going to take more or less 40 to 45 minutes. And then we're going to have some 10 to 15 minutes at the end for Q&A, depending how long the code takes to run. But that's more or less how long it's going to take. But for the README file here, I have set up all of the instructions that you guys need to run sequentially. If you'd like to clone the repo and then run this on your machine locally, or if you like to use a GPU on the Intel Tiber AI Cloud. So I'll be walking through that as well. Not super in depth, but just let you guys know that if you'd like to use a GPU and it's completely free, you can go on Intel Tiber AI Cloud and you can use a GPU from there, which is where I'm going to be running all of the code today. So if you just go over, I believe there's 6 steps to set up the repo. And then this last step here at the bottom, you only need to do it if you are running on Intel Tiber AI Cloud. So to get started with the hands-on portion of the webinar, the first thing that you're going to do if you like to run this on Intel Tiber AI Cloud on the GPU, you're going to go to Intel cloud.com or cloud.intel.com, cloud.intel.com. And then you're going to sign up with an account for yourself. I already have done that. And then the next thing you're going to go on Learning Catalog notebooks. And then there's going to be, if you scroll down here a little bit, let me make my screen a little bigger. You're going to see that there's a PyTorch on Intel GPUs. And then you're going to click Launch. And if you click Launch, it's going to open up. I already have it open. But it's basically going to open up this Jupyter instance where you're going to have access to Intel GPUs on data centers, which they're basically called XPUs for PyTorch. And we're going to be leveraging PyTorch for basically everything that we're going to be doing today. And that's it. So then you just go through the steps by steps that I have on the README, and you'll be able to set up your environments. So I already have cloned the repo here. As you can see on the terminal, I already have passed the tests that it's on the README. Is one of the steps. You just run tests to check that the environment is set up correctly. So I already have done that. I'm not going to go through that again. But to get started, basically, we're going to be covering 3 main notebooks today in the webinar. So if we go to the notebooks folder, there's going to be an EDA notebook, so exploratory data analysis. There's going to be an embedding notebook, and also a FISE notebook, which is a similarity search that we're going to be using with the embeddings. So, and we're going to be running those sequentially. So we're going to start with the EDA notebook. I already have it open here. Make sure that if you are running this on the Intel GPU instance here on Jupyter, that you have the PyTorch 2.6 kernels selected. Otherwise, you're not going to be able to use the GPU to run this. So to get started, this EDA, or this exploratory data analysis notebook, it's really about understanding the domain shifts in the PlantCLEF competition. So like I said, all of the training data is single label, and all of the test data is multi-label used in the competition. So there's a huge domain shift between the training data and the test data. And our goal is really to, on this notebook here, is to better understand and kind of get a feel for that data distribution. And I think one thing worth noting is that not only that's the main challenge of the competition, the big difference between the single label to the multi-label, but also the size of the data. So just so you guys know, today in the webinar, we're going to be working with a subset of the training data and a subset of the test data. The training data has 1.4 million images total. So again, I'm just going to go back here to the Kaggle web page, but if you go to the data tab here, you'll be able to see the sizes of the data sets that are going to be available to you. So here's the test images, yeah, some metadata CSV and so on. But the test data has 2,100 images, but the training data has 1.4 million images total. And I believe they mentioned here somewhere, but you can see that the training data is almost 300 gigabytes in size. And yeah, they mentioned right here, 1.4 million images for the training data. So it's quite a large data set. So not only we're trying to bridge the gap of this domain shift between the training data and the test data, but we're also dealing with a lot of images. But for this workshop, so for this webinar, so we run things in a timely manner, we're just going to be working with a subset of the data. So I'm just going to be starting to run the code here. We're just importing some libraries, we're importing device. If you're running this on the GPU on Intel, you definitely want to see that you are using the XPU here, the 2.6.0 XPU. Again, here we're just importing some dependencies like pandas, since we're going to be working with that. And we're going to be reading the data. So I already have, if you go through the README file, one of the steps is going to be downloading the data. I have the subsets in a Google Drive, and you basically just run a bash script, and you'll be able to download the data to your repo locally. So if you go here, just to show you, there's going to be a data folder. Under the data folder is going to be a Parquet folder, and under the Parquet, there's going to be a subset of the test and a subset of the train data. So I'll go back here to the notebooks. So if we run this out, basically we're just reading the data and then checking that everything is correct. So taking some time to look at this, the training data has, the subset of the training data that I selected has 2,020 rows by 22 columns. Some of the most important columns that we have here is the image name, which is unique to each image on the training set. Also, the data is being stored as binary formats. That's why we see this kind of weird looking string here. That's just binary. And then we also have another very important column is the species ID, because that's actually the predictions that we're going to be generating. We're going to be looking at the images and trying to predict their species, which is going to be basically the species ID. And then we have all kinds of metadata as well, like the altitude, longitude, I believe as well. We have the species, the name of the species, the genus, and so on and so forth. We even have the links, the URLs of the images. So if you'd like to check that out, you can. And then for the test data is a subset of those 2,000 images. I selected only 200 images out of the 2,000. So the test data is going to be only 200 images, and there's only 3 columns. It's going to have the image name, the path where we extracted the images, and then the data, which is also in binary formats. So the next cell, I prepared some very neat visualization functions here. So this one, we're just plotting some of the images from the training data, and just to take a look at them to see how different they look like. So if I kind of zoom out here a little bit, we have this 3x4 kind of grid of images, but each one of them is a different image on the training data. As you can see, there's only one species per image. So this one right here, I believe we have 12 different species. And yeah, we're just taking a look at the images, but you can see that they vary greatly. The backgrounds are different. Maybe the time of the year that they were taken is also different. And like I said, the entire training data, it's 1.4 million images. We're just working with 2020 here as a subset, but yeah, it's quite a lot of images. So let me zoom back in again and close this. So here, we're just going to take a look at the multi-label images on the test data to see how different they look like from the training data. So this, I'm just selecting some interesting images here on the test data. There is only 12 images that I selected, and then we can plot them as well, just like the other one that we did with the single label training images. So if I also zoom out here a little bit, you can see how difficult this challenge is, because how different those images on the test data are. As you can see, they vary greatly from one another. There's all types of not only plants, but also flowers, but there's also dirt, like things that we don't want to identify. And you can see that they look quite different from the training images. And that's a huge challenge, right, in machine learning. You want to have a good representation of your data that you're making predictions. So basically, we're just exploring some of those images, seeing how they differ. And then here, the next one, it's basically I'm going to be showing you the main approach that we're going to be doing today. So basically, the baseline approach for this competition, perhaps the simplest thing that we can do, is to crop the image into tiles. So in this case, this is one of the test images, just as an example. And we're going to be cropping the test image into this 3x3 grid of tiles. So we have this tile 1, it's like this part here from the image. And then the second tile, it's somewhere over here. And then the third tile, it's right here. And you can see so on and so forth. We're basically cropping and chopping the image into those different tiles. And then we're going to be classifying which species are in each of those tiles. And we're going to aggregate them in a clever way. And then we're going to be able, to address that domain shift between the training data and the test data. So this is the approach that we're going to be doing today for the workshop, or for the webinar. I keep saying workshop. And here I'm just showcasing you guys that we only have 200 images on the test data. So that's basically the EDA notebook. Very simple, just taking a look at some of the images. The next notebook that we're going to be running, and I'm going to close the EDA one, is going to be the embedding notebook. So basically, I kind of explained to you the main thing that we're going to be doing. We're going to be cropping the test image into tiles, and then we're going to be doing something with those tiles. So in this notebook, we're going to be basically focusing on this feature extraction pipeline that we have here, the one in blue. So basically what we're going to be doing, the organizers of the competition of PlantCLEF, they made available a vision transformer model that is called VIT-D2PC24all. And yeah, quite a long name, but it's basically a vision transformer model that was pre-trained using the DINOv2 method. So DINOv2 is a method, a self-supervised method for training vision transformers. And those are types of neural networks. And then this model specifically was later fine-tuned using the 1.4 million images of the training data. And the organizers make that available for us. So anybody that wants to use this model is available in the competition itself. And basically what we're going to be doing, we're going to be extracting embeddings, which are high-dimensional vectors, from those images, not only from the single label images, but also on the multi-label images. And then we can do all sorts of things with those embeddings. We're going to be using them later on in a downstream task to basically use transfer learning and utilizing FISE, which is a nearest neighbor search algorithm. FISE is a library that was created by Meta. DINOv2 was also created by Meta. So basically, we're using libraries that were created from Meta today. And we're going to be using FISE to basically look at the embeddings and look at the images that are most similar to the tiles. And we're going to be doing classification that way. But for the embedding notebook, that's what we're going to be doing. So let's start just running some. Oh, what I'm going to do, actually, I'm going to restart and clear all outputs. I'm going to make sure that I'm running on PyTorch 2.6 here. And I'm going to be running the cell. So again, we're just importing some libraries, and then we're importing the data just like we did in the previous EDA notebook. The next thing that I'm going to be doing, just to showcase how this is working, or just showcasing the pipeline, I'm going to be limiting the data to only 2 images, so it runs faster. But that's it. So the first thing that we're going to do, I created this torch_pipeline function, which I'm going to show you in detail what that's doing. But for now, just assume that basically what we're doing, we're passing the images through the model, and we are extracting not only the embeddings for the images, but we're also going to be extracting their logics, which is basically the confidence of the model for what species are going to be in each image. And for we're going to be doing this on the training sets, only 2 images, since we want to run this fast. And you see that as I run this cell, is going to pop up the TQDM process. It's done very fast. And then we check the shape. So basically, the shape that we have, we just ran through 2 images. Each one of them is only 1 tile because we're not cropping the training image right now. And each embedding is going to be a 768 dimensional vector. So it's going to be a vector with 768 different floating point numbers. And then we can check the first 100 values of those 768 numbers. So here, as you can see, it's just a bunch of floating point numbers. Another thing that we can do is create a data frame with those embeddings. And then again, I also created a bunch of different plotting functions to showcase the images, but also the embedding. So the 2 images that I selected were those ones, basically the first 2 images on the training data. And then I can plot their embeddings as well. So basically, this is what the computer is understanding. To us, it doesn't have any meaning, but each one of those are the 768 different floating point numbers in a grayscale plot. So basically, this image right here creates this embedding, and this image right here creates this embedding. So this is what we extracted. Basically, the embeddings, they are a lower dimensional vector or lower dimensional representation from the image. So basically, the model was able to extract the features of the plants and basically encode them into this vector representation, as we see here. And then now we're going to do the same for the test data. But for the test data, we're actually going to be chopping the image into those grid of tiles. And we're going to be extracting the embeddings and also the top K logic. So for each tile, we're going to be extracting their embeddings. So the vector with 768 floating point numbers. But also we're going to be extracting basically the top 5 species, because the model, the DINOv2 model that we're using here, the VAT, also has a linear layer on top of it, and we can make classifications using the model. So we're going to be doing that here. And to create this 3x3 grid of tiles, the one thing that we're doing, we're also limiting the test data to 2 images. So I'm grabbing those 2 images, that's the image names. I'm filtering those images into a new data frame, and then I'm passing them through the same pipeline. I'm just passing those images into the model. The data pipeline is going to be cropping the image into those tiles, and then it's going to be feeding each tile into the model, and the model is going to be outputting not only the embedding, but also the logics, so the species classification of it. And if we check their shape, is going to be different. So for the training data that we saw previously, it was 2 by 1 by 768. This time, because we're doing this grid of tiles, is 2 by 9 by 768. So where is the 9 coming from? The 9 is just because we have a 3 by 3 grid, it creates 9 tiles. So basically this says that each image is going to have 9 different tiles, and each of those tiles is going to be an embedding with 768 dimensional embedding. And then here we're going to be basically is a function to be exploding those embeddings. So we're going to be passing a data frame. In this case is the test image data frame that we created with only 2 images. And we're going to be creating this data frame with not only the embeddings, but also the logits and the tiles. So for this image, you can see that is the same image for all of them. So it's one image created 9 different tiles. In programming, the indexes start at 0, so 0 through 8, it's 9 different tiles. And for each one of those tiles, we have the embeddings, and we also have the logits. So we have the species, and then this right here, this number, it's basically how confident the model predicted that it was this species. And because we're taking the top K species for each tile, we're taking basically the top 5 species for each tile. And we get not only their species ID, but also their predictions, basically how confident the model is that we are predicting that. And we can plot them here as well. So again, just to reiterate, that's kind of what we did for the test image. So we selected this image, we cropped the image into those 9 different tiles, and then we can also visualize their embeddings. So this is what each embedding will look like for each tile. And here we have another plotting function. We're just plotting the embeddings on their own. So you can see that, for example, this the last embedding here, number 8, because we're starting at 0, is going to be corresponding to this one here. Just the indexes are different, but they're basically the same embeddings. And here we're going to be classifying. I've been talking a lot about the embeddings, but going back to the logit. So for, like I said, each tile, we look at the top predicted species and we associate them, their scores with their scores, which are the logits. So yeah, this is just gonna help us understand like what it's doing. So basically, again, we only have 2 images because we basically filtered down to only use 2 images for this data set. For the first tile, like I said, because we're selecting the top K, in this case, K equals 5, we're only selecting the top 5, the top 5 predictions per tile. And this is how confident it is in a descending manner. So it predicted that this species was 8% confident, and this species was 5% confidence. And I have to, just one thing that I'd like to note is that those predictions are out of the entire training data, and the training data has 7,800 species, and the test data has about 800 species. So the test data has 1 order of magnitude more species than the training data has 1 order of magnitude more species than the test data. And then here we can see all of the logit predictions that we generated for each tile. So if we just want to use the fine-tuned model to basically predict species, we're fine. We don't have to use the similarity search algorithm that we're going to be running later on. But yeah, like we can get predictions, not only the embeddings, but also the logits. And then the next thing that we're going to be doing is, right now, we only embedded the 2 images on the training set, and we also embedded 2 images on the test set. We created the grid of tiles and we embed those. But we actually want to embed the entire training set and also the entire test set. So what we're going to be doing here right now is we're going to be embedding the entire test sets with this tiling approach. So instead of doing that with the 2 images, we're going to be passing the entire test set to this torch pipeline function. The batch size is going to be 10. So each batch that we're passing through the model is going to have 10 images. Out of those 10 images, because we're doing a 3x3 grid, is going to create 9 tiles for each of those images. So each batch is going to actually have 90 tiles, so 9 times 10. And yeah, we're just going to be passing those parameters through this function. And while this function starts running here, I think I'd like to take some time to actually show you guys, since this is a PyTorch webinar, how we're actually using PyTorch for this. So in the repo, you will notice that we have a few folders. So the notebooks are going to be under the notebooks folder, but there's also this PlantCLEF folder. And this one is really important. So there's going to be the embed folder, which is going to have this workflow.py file. I'm going to talk about that actually right now, because that's what we're running. And then later I'm going to be talking about the model and how we're actually extracting the embeddings. But basically, this function right here, torch_pipeline, I'm importing this function from this workflow.py file right here. And basically what this is doing, what this function is doing, is that we are initializing the DINOv2 Lightning model. So I'm using PyTorch Lightning to initialize the model and extract the embeddings. But we're also creating a plant dataset and data loader, which I'm going to be talking about both the model and the dataset later in the webinar, like maybe when we embed the training images. But basically, we are just initializing those classes that I defined on different files. And then all this is doing is that we're just extracting both the embeddings and the logits. And then if we set the parameter use grid equals true, right now it sets to false, but if we set equals true, which we did for the tiling approach for the test images, is going to chop the image into those grid of tiles, and is going to do all of that in the plant dataset class that we have. So let me go back here. So it's done. It basically took almost 2 minutes to embed the entire test sets, but just keep in mind that we're using only 200 images in this case. But because we are doing this grid approach, as you can see here, just checking, we have 200 images. Each of those images has 9 tiles, and each one of those tiles has a 768 dimensional embedding. And then we're going to be exploding them to create a data frame. So this function right here is already defined. And you can see, because we have 200 images and each one of those images generates 9 tiles, we actually have 200 times 9 is 1800. So you're going to have this data frame with 1800 rows by 5 columns. And 3 of those columns that were created were the embeddings column, which is the embeddings that the model extracted and generated, the logits column, and also their tile numbers. So basically what we did previously, we did again, but in a much bigger scale. And here we're just plotting, for example, the first image on the test data, and we're just checking the image itself and the tiles, the embeddings that were generated for each tile. And then the next thing that we want to do is that we want to save this data set that was created so we can work with the data sets later in the FISE workflow for the similarity search algorithm. So we're going to run this. And to show you guys, we have here on the repo, we have a data folder. And basically what this is going to do is that you see this as it's still running, but once this finishes running, it's going to be creating this folder called embeddings, and then the name of the folder name that we're storing the Parquet embeddings is task grid 3x3, which is the grid size embeddings, and this is going to be basically just Parquet files of the data. So instead of storing those embeddings in like a CSV, we're just storing them in Parquet because it's a more efficient way of storing data. And the next thing that we're going to do is we're also going to be embedding the training images. In this case, the training images, if you guys remember, there are 2020 images on this subset of the data. The entire data has 1.4 million, but I selected a subset of that, which is 2020 images. And if we run this, again, this is going to take about 2 minutes to run, to embed all of those training images. But to show you guys what we're doing and how we're utilizing PyTorch, if you go under the Torch folder, there's going to be two Python files, a data file and a model file. If we go into the data file, this is where we are creating that plant dataset class, which does the grid process into processing the test image into the grid of tiles. If we set the use grid parameter equals true. If we don't set equals true, if we keep it as false, then it's not going to do, it's not going to chop the image into a grid of tiles. It's just going to embed the entire image, or basically process the entire image so it can be embedded by the model. And yeah, if you'd like to check it out, this is basically the function that we're doing this, and we're using PyTorch to do all of this. There's also a Lightning data module function here at the bottom. Also, something to notice, I guess, just to mention very quickly, is that there's this torch_pipeline function, but there's also a PyTorch Lightning function that you can find in the same embed workflow. If you're running this on another environment, like on macOS, or if you're running this on a NVIDIA GPU, then not only you can use the straight up PyTorch pipeline, but you can also use the PyTorch Lightning pipeline for this that I also created. And then the other file that I wanted to talk about is this model file, which is where we are creating and instantiating the DINOv2 Lightning model. So here, basically, there's a bunch of setup that we do beforehand that comes from other files in the PlantCLEF folder. But basically, what we're doing here, we're loading the class mapping, so the classes of plant species IDs to do the classification on the forward pass. So for each batch, we are basically just extracting the embeddings. And I'd like to point out that the embeddings generated by the VIT model are actually much more high dimensional. In our case, we are extracting the CLS token out of those embeddings. So the CLS token is basically just the last dimension in the outputs generated of the embeddings, and we are extracting the 768 vector of that outputs. And then for the predict step, that's where we actually generating the logits, where we are predicting the species on each tile for the test data, and also for the training data as well. So if we go back here, that's basically what this pipeline is doing. So we can keep running this. Again, the pipeline, the training data, the subset has 2020 images. Each one of those embeddings has the same dimension. And then here we are exploding the embeddings. Again, the same function that we used before. You see that all of the tiles are equal 0 because for the training data, we're not chopping the image into tiles. We're just embedding the entire image. And we get the prediction. So we get the embeddings, we get the logits, and we're going to be using this later in the other notebook for similarity search. So if we keep running here, again, another function just to check. So we have the single label image on the left, and then on the right, we have the embedding that was generated for the image. And this right here is the image name, so you can see that they're the same. And then the same thing that we did for the test data, we're going to be saving that into Parquet, into the data directory here that we created. And then that's it. So basically, we're able to run this embedding workflow. Now we have some embeddings that we can use for downstream tasks, like using a similarity search algorithm to classify the titles. So those two last cells here on the bottom, we're just checking that the data frames were created successfully. So we're just reading them and showcasing the five first rows into the data frame. And that's it. Like, we have embeddings and we can use them for classification into a downstream task. So for the last notebook of the webinar today, is going to be this FIS notebook. FIS stands for Facebook AI Similarity Search, and it's a library that was created by Meta that focuses on similarity search. So basically, the previous notebook, we did this blue pipeline here. Now that we already extracted the embeddings from the single label images and also the embeddings from the multi-label images, or all the embeddings for the tiles, we're going to be using those tiles, or we're going to be using those embeddings into a transfer learning approach into this nearest neighbor search algorithm, extracting their top K probabilities and then outputting that as our predictions for the competition. So basically, this notebook right here is all about similarity search, and we're going to be leveraging the self-supervised feature representations, which are the embeddings, to classify the test images. So again, I'm going to make sure that I'm on PyTorch 2.6. I'm going to be running. Actually, I'm going to be, let me do restarts and clear out outputs so we can run one by one. I'm going to be running all of the cells. Again, here we're just importing PyTorch. We're importing pandas. And then we're importing the data. And here, just to note, we start planning. Okay, let me run that again. Run. Yeah, so we're going to be loading the embeddings that we created on the previous embedding step. So the data frames that we created and we wrote to the data path, we're going to be reading those data frames here, and we're going to be using those embeddings to do similarity search so we can classify each tile. So here we are. PlantCLEF appears to have died. Okay, it died again. Let me do clear output cells. Select. Okay, let me try to run that again. Import, good. Imports data. Come on. Yeah, let me reload the page. Okay, restart. Hopefully I'll be able to run this. So again, here we are loading the data. Okay, I guess I'm not going to be able to start this notebook. So what I'm going to do, I already have this notebook ran. Let me check something. CD, get status, get restore, notebooks, files. So let me close this Discord. Okay, so I'm not going to be running this. I already ran this beforehand, so we can see the outputs here. But basically what we're doing, we're loading the embeddings that we created in the previous step. I don't know why the kernel kept restarting, but it just did. And then here we're going to be performing the similarity search using FIS. So we're going to be importing this FIS classifier class that I also created. I'm going to show you very quickly what it's doing and where you can find it. But basically, it's under PlantCLEF, the FIS folder, and the classifier.py file. And basically what is this doing is just performing similarity search with the training embeddings and for each tile on the test embeddings. And it's basically finding which embedding on the training data matches the closest with each tile on the test data. And it's doing so by using cosine similarity, which is just an algorithm to find distances between high-dimensional vectors. And so if I go here on PlantCLEF and we go PlantCLEF folder, files, and then classifier. So this is going to be the files classifier class. And there's only two, it's very simple. There's only two methods within the class. This one is going to build the files index from the training data. So it's going to take the embeddings on the training data and it's going to index them, basically performing inner product, which is the dot product of those embeddings, and then storing them, their indexes. And then the make predict, we're going to be passing the embeddings from the test data, and then files is going to search into the index that created and find the embedding that matches the closest with the test data. So we do that here. We pass k equals 1, so we're just trying to find the one embedding that fits the closest with each tile. And so for 5 tiles, for example, we have those images right here, and those are their similarities. So how confident FICE is that it predicted those images for each of those tiles. Here we do the same, but we increase k equals 5. So for each tile, we're grabbing the 5 most similar embeddings from the training data. So we can do more multi-label classification and increase our confidence that that species is actually present in each of those embeddings. And then we get a different shape. So for the test data for all of the 1,800 different images that were generated by tiles, we have this shape right here. And then here we're just creating the data frame that was created. So it's basically a similar step from the exploding function that we did for the embeddings. We create this data frame that has the image name, it has the data in binary, it has the embedding, the logits, but also has the predictions that were generated by FICE, and also the similarities. This is basically the confidence score for each prediction. And we also have the species ID, which were the predictions of the species that FICE created. So again, for this image specifically, we chop the image into those 9 tiles. And then here we're just visualizing the prediction. So for the tile, for the first tile, it predicted those 5 different images. You can see that the first 4 of them are the same species ID. And then the last one is a different species. And you can see that we do that for every single tile in the test data. So for each tile, now we have 5 different predictions. You can see that this one as well, the first 4 predictions are the same species, and the 5th one is a different species. So by doing so, you can see that we're able to kind of tackle this multi-label challenge by predicting not only one, but maybe more than one species on the test data. And then basically how FICE is working is that we're performing this k nearest neighbor search on the image embeddings, and it's going to be finding the top k training embeddings that are most similar to each test tile. And each training image has a single image species that we want to predict. And then we're going to be mapping those predictions back to the species. So here you just have an example of 5 predictions of the first tile. As I said, the first 4 are the same species. They're different images, but they're going to be the same species. And the 5th tile is a different species. So it basically predicted 2 species on that single tile. And then at the end, we create this submission CSV file that you guys can use and submit to the competition. Just note that because we are dealing with a subset of the test data here that only has 200 images, the real test set has 2,100 images. So if you try to run this workflow and submit to the competition, it's not going to work. But that's pretty much it for the webinar. before I close, I'd like to just point out that this competition goes way beyond PlantCLEF. There's also FathomNet, FungiCLE, and GeoLifeCLE, and those are different, basically biodiversity problems that we're trying to solve using machine learning. So if you like biodiversity, if you like machine learning, if you're new to deep learning, I think those are an amazing way to get started on your machine learning or programming journey and really have fun. And this does really go out to say that, today there's a lot of use cases being done with AI. We hear a lot of things about LLMs, but there's also really amazing things that we can do with AI, like tackle those biodiversity conservation challenges, which I'm very passionate about, as you can tell. So I highly suggest, if you're a beginner or intermediary level, or even advanced, to consider competing on those competitions, because then you can post your results and you're actually helping researchers in some way advance this field and try to use AI for a positive impact. So yeah, with that, that's basically it. Feel free to check out the GitHub repo. If you have any questions, please feel free to reach out to me as well. But I believe that we can open up for Q&A.
Okay. All right. Thank you, audience, for your great questions. Murilo, I have a lot of questions for you. So let me ask this first question. And I'm trying to note where the question was so you have the context for it. So while you were working in the embedding notebook, there was a question. This is a multi-part question.
Okay.
Are you cropping the pics so you can augment the data set? The second part of that, is this only for the test data set? And do you do this to have more confidence on how the model performs?
Okay, let me address one by one. If I'm cropping the pixels, the first one,
Yeah,
are you cropping the
augment?
pixels? Okay, so you can augment the data set. That's a great question. So for the demo here that I did today on the webinar, no, I'm not cropping them. I'm cropping them only to plot them into the visualization. For the competition last year, we did crop the training data, the entire training data, to 128 by 128 into dimension. And only by doing that, we're able to reduce the size of the data set from the 281 gigabytes all the way down to 15 gigabytes. So much more manageable, right? The second question, is this only with the test data? So for the test data, the reason why we're cropping the test data is because if we try to classify the entire image, like doing this approach, is gonna miss a lot of detail. And also, something very particular about the model that we're using, the model basically it's best used with images that are roughly 500 by 500 pixels in size. And because the test data has almost all of the images on the test data, they have like 2000 by 2000 pixels. If you do a grid of 4 by 4, you kind of have each tile having those 500 by 500 pixels. And it works really well for extracting those embeddings. And that's just something specific with the model. And then for the third question, do you do this to have more confidence? Yeah, so basically we do that to have a more confidence scores on each of those tiles. Like you saw for one of the tiles, or I think it was for more than one of the tiles on that example, the first 4 predictions using phy's were the same species. So the similarity search algorithm was very confident that that species was on that tile. And yeah, so basically those are the answers.
Great. So I have 3 more questions on this particular notebook. Does it use BERT's CLS token to get the image embeddings, or is it a different architecture that's being used?
Yes, it's a different architecture. It's not BERT, but we do get the CLS token. And that's something that I covered. The person probably asked the question before I mentioned the CLS token, but we do get the CLS means classification. So we get the classification token from the embeddings that were generated by the VIT. And yeah, so we are getting the classification token.
Okay. Next up, what are the logits? Where do they come from?
Okay, so the logits are basically just confidence numbers that the model generated for each tile. So basically, where did they come from? This model, specifically the VHC that I'm using, not only it's an encoded model, which you can encode and get embeddings from it, but it also has a linear layer on top of it that you can perform classification. So when you perform classification on each one of those images, you get the classifications, which are the species ID. But not only you get the species ID, but you also get a confidence score of how certain the model is that species. And you get a score for each species that are in the training data. Because the training data has 7,806 different classes, you get, for each image, you have 7,806 different predictions, each one of them having a classification between 0 and 1. So that's basically a confidence of the model. So basically, what are the logits? They're just a confidence of how the model is classifying the images.
Okay, and the last question while you were working in the embedding notebook is top K classes a hyperparameter, and how do we know a specific image has that many species of plants?
Okay, that's a good question. It's not a hyperparameter because we're not training the model in any way. It's just for each tile, the top K only works if you're doing the grid approach, where you are extracting the tiles from the images. So for each tile, the top K, it just determines how many logits you are extracting. So you can extract, in that case, I was only getting the top 5, but I could get the top 7,806, which would be all of them. The reason why I'm getting 5 is because I know for a fact that an image is not going to have more than like 10 species. I think that's kind of the upper limits that we see. So if I'm getting the top 5 species for each tile, and if I do a 3x3 grid, at the end of the day, I'm going to have 45 total predictions because it's 5 for each tile. I have 5 tiles, 5 times 9, 45. And then a lot of them are going to be repeated. So we kind of get their sets. We eliminate the ones that were duplicated, and we only get the ones that were highly confident. So that's basically what we're doing. So it's not a hyperparameter. It's just something that you can pass bigger numbers, like you get the top 10 or the top 20. You're just going to have more predictions. In that case, I just did the top 5.
Okay, and I'm going to go ahead and ask you the last question because we only have 2 minutes, and I think this is really important. And this is when you are working in the FEISS notebook.
Okay.
Why use FEISS and not some other vector DB search?
Okay, yeah, no, that's a good question. Why use FEISS? FEISS is just a great library for finding and doing similarity search. It's really simple to use. The way how it works is that it looks at each, in our case, it's gonna look at each individual tile, and doesn't really consider how tiles relate to each other to like the broader sense. So it has some built-in, very clever techniques into that. When we were doing the FEISS for the entire training data set, like the 1.4 million images, it took only a couple minutes to run it to create the index because it has a lot of efficiencies built on the back end. And it's just like a library that is well known in that space. Like other researchers have done and have used FIs for that. So yeah, it's just something that it's easy to use, it's reliable, it's well maintained, it's reproducible, which is something that you want to have in research, and it works really well.
Great. Well, that's audience. That's all the time we have today. Thank you so much for joining us, Murilo. Thank you so very much for sharing your work with us. As always, follow PyTorch. There will be other webinars, blogs, events. Thank you all very much.
Yeah, thank you.